Dear viewers, this video is part 3 of the 7-part series on lymphocytes and immunity in the Medicine for Anyone in the YouTube channel. Role of thymus. The thymus is an organ that is critically important to the immune system, which serves as the body's defense mechanism, providing surveillance and protection against diverse pathogens, tumors, antigens, and mediators of tissue damage. The immune system comprises a complex network of cellular and molecular components subdivided into thymus independent, that is innate immunity, and thymus dependent adaptive immunity, the both arms which function synergistically in all immune responses. Innate immunity constitutes the first line of defense and is mediated by innate immune cells such as tissue macrophages, dendritic cells DCs, and granulocytes which elicit their effector function within minutes to hours following antigen exposure. Innate cells become activated via germline encoded pattern recognition receptors PRRs, including toll-like receptors TLRs and NOD-like receptors NLRP, which recognize invariant features of pathogens, pathogen associate molecular patterns PAMPs, and tissue damage. Note, NOD-like receptors NLRs, nucleotide oligomerization domain, that is NOD-like receptors NLRs, are a specialized group of intracellular proteins that play a critical role in the regulation of the host innate immune response. Once activated, innate cells such as macrophages and neutrophils can effectively clear antigens via phagocytosis. Other types of innate cells such as DC, dendritic cells, take up and process antigens, resulting in expression of antigenic epitopes in conjunction with their major histocompatibility complex of human leukocyte antigen molecules. These dendritic cells can then serve as antigen presenting cells APC for the priming of the adaptive immune system. In this way, the early innate response is coupled to and facilitates adaptive immunity. The adaptive immune system consists of T and B lymphocytes which express specific antigen recognition receptors and develop highly specialized effector functions with the ability to form long-term immunologic memory. Both B and T cells develop from bone marrow-derived progenitors. While mature B cells are exported to the periphery directly from the bone marrow, T cell development, maturation, and export require critical differentiation steps to occur in the thymus. Thymus-dependent T cell differentiation processes include the expression of an antigen-specific cell surface T cell receptor TCR through recombination of germline encoded gene segments and thymic education involving negative selection, that is elimination of potentially self-reactive T cells, and positive selection of T cells with the capacity to recognize antigens encountered in the periphery. These important thymic processes ensure that T cells can recognize antigens in the context of self-MHC, that is major histocompatibility complex, but do not elicit reactivity. Once exported from the thymus, the resultant naive T cells populate multiple secondary lymphoid organs, including spleen and many lymph nodes. Dear listeners, by the way, this educational video is made possible by PrescribeOn, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribeon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. One thymic lymphocyte develops specific reactivity against one antigen, and then the next lymphocyte develops specificity against another antigen. This process continues until there are thousands of different types of thymic lymphocytes with specific reactivities against many thousands of different antigens. These different types of pre-processed T lymphocytes now leave the thymus and spread via the blood throughout the body to lodge in lymphoid tissue everywhere. Mature T lymphocytes constitute 70 to 80% of normal peripheral blood lymphocytes, constituting only 2% of the total body lymphocytes contained in peripheral blood. 90% of thoracic duct lymphocytes, 30 to 40% of lymph node cells, and 20 to 30% of spleen lymphoid cells. In lymph nodes, T cells occupy deep paracortical areas around B cell germinal centers, and in the spleen, they are located in periarteriolar areas of white pulp. T cells are the primary effectors of cell-mediated immunity, with subsets of T cells maturing into CD8 positive cytotoxic cells capable of lysis of virus-infected or foreign cells, that is, short-lived effector T cells, and CD4 positive T cells capable of T cell help for CD8 positive T cell and B cell development. Two populations of long-lived memory T cells are triggered by infections: effector memory T cells and central memory T cells. Effector memory T cells reside in non-lymphoid organs and respond rapidly to repeated pathogenic infections with cytokine production and cytotoxic functions to kill virus-infected cells. Central memory T cells home to lymphoid organs where they replenish long and short-lived and effector memory T cells as needed. In general, CD4 positive T cells are the primary regulatory cells of T and B lymphocytes and monocyte function by the production of cytokines and by direct cell contact. This figure shows the immune system regulation. On specific stimulation by processed antigen, the T helper CD4 cells produce lymphokines which activate cytotoxic CD8 positive T killer cells, stimulate suppressor T CD4 cells at the appropriate time, and stimulate B cells to modify themselves to plasma cells to produce various types of immunoglobulins. See figure immune system regulation shown earlier. In addition, T cells regulate erythroid cell maturation in bone marrow. Through cell contact CD40 ligand, they have an important role in activation of B cells. Colonization of the gut by commensal bacteria, the gut microbiome, is responsible for expansion of the peripheral CD4 T cell compartment in normal children and adults. Self and non-self development. The thymus also makes certain that any T lymphocytes leaving the thymus will not react against proteins or other antigens that are present in the body's own tissues. Otherwise, the T lymphocytes would be lethal to the person's own body in only a few days. The thymus selects which T lymphocytes will be released by first mixing them with virtually all the specific self antigens from the body's own tissues. In a if a T lymphocyte reacts, it is destroyed and phagocytized instead of being released, which happens in up to 90% of the cells. Thus, the only cells that are finally released are those that are non-reactive against the body's own antigens. That is, they react only against antigens from an outside source, such as from a bacterium, toxin, or even transplanted tissue from another person. Most of the pre-processing of T lymphocytes in the thymus occurs shortly before the birth of a baby and for a few months after birth. Beyond this period, removal of the thymus gland diminishes but does not eliminate the T lymphocyte immune system. However, removal of the thymus several months before birth can prevent development of all cell-mediated immunity, including rejection of transplanted organs. The thymus exports about 2% of the total number of thymocytes per day throughout life, with the total number of daily thymic emigrants decreasing by about 3% per year during the first 4 decades of life. Liver and bone marrow pre-process B lymphocytes. In humans, B lymphocytes are pre-processed in the liver during mid-fetal life and in the bone marrow during late fetal life and after birth. B lymphocytes are different from T lymphocytes in two ways. Instead of the whole cell developing reactivity against the antigen, as occurs for the T lymphocytes, the B lymphocytes actively secrete antibodies that are the reactive agents. These agents are large proteins that are capable of combining with and destroying the antigenic substance. 2. The B lymphocytes have even greater diversity than the T lymphocytes, thus forming many millions of types of B lymphocyte antibodies with different specific reactivities. After pre-processing, the B lymphocytes, like the T lymphocytes, migrate to lymphoid tissue throughout the body, where they lodge near but slightly removed from the T lymphocyte areas. We have already seen the diagram of a lymph node and all the different structures in it. Especially concentrate the attention on the B cell and T cell zones in the cortical and paracortical areas. See the functional diagram of a lymph node shown earlier. Note, T follicular helper T cells, TFH, CD4 T cells in B cell follicular germinal centers that produce IL-4 and IL-21 and drive B cell differentiation and affinity maturation in peripheral lymphoid tissues such as lymph node and spleen. Th1 T cells. These are T4 CD4 helper T cell subset participating in cell-mediated immunity. Th2 T cells. CD4 helper T cell subset produces IL-4, 5, 6, 9, 10 and IL-13 and regulates antibody and eosinophil responses. T regulatory cells, Treg, CD4 and CD8 T cells, regulated by the transcription factor FOXP3 that play roles in modulating the immune system to prevent deleterious immune reactivation. Th9 T cells, that is CD4 T cells regulated by the transcription factor PU1 that secrete IL-9 and enhances inflammation in atopic disease and inflammatory bowel disease as well as mediate anti-tumor immunity. Th13 T cells are T follicular helper cells regulated by the GATA3 transcription factor that produces IL-4, 5, 13. Th13 TFH induces high affinity IgE antibody responses that causes anaphylactic reactions to allergens. Th17 T cells, these are CD4 T cells secreting IL-17, 22 and 26, playing a role in autoimmune inflammatory disorders as well as defend against bacterial and fungal antigens. See the figure functional diagram of a lymph node showing B cell and T cell zones again. T lymphocytes and B lymphocyte antibodies react against specific antigens, the role of lymphocyte clones. When specific antigens come into contact with T and B lymphocytes in the lymphoid tissue, some of the T lymphocytes become activated to form activated T cells and some of the B lymphocytes become activated to form antibodies. The activated T cells and antibodies in turn react highly specifically against the particular types of antigens that initiated their development. The mechanism of this specificity follows. This picture shows an antigen activating only the lymphocyte having complementary cell surface receptors and recognize a specific antigen. Millions of different clones of lymphocytes exist, shown as B1, B2, B3. When the lymphocyte clone B2 in this example is activated by its antigen, it reproduces to form large numbers of duplicate lymphocytes which then secrete antibodies. See the figure, antigen activating only the lymphocytes having cell surface receptors that are complementary and recognize a specific antigen, shown just earlier. Millions of specific types of lymphocytes are stored in lymphoid tissue. Millions of different types of preformed B lymphocytes and preformed T lymphocytes capable of forming highly specific types of antibodies or T cells are stored in the lymph tissue as explained earlier. Each of these preformed lymphocytes is capable of forming only one type of antibody or one type of T cell with a single type of specificity, and only the specific type of antigen can activate it. See the figure shown above. Once the specific lymphocyte is activated by its antigen, it reproduces wildly, forming tremendous numbers of duplicate lymphocytes. If it is a B lymphocyte, its progeny will eventually secrete the specific type of antibody that then circulates throughout the body. If it is a T lymphocyte, its progeny are specific sensitized T cells that are released into the lymph, carried to the blood, and then circulated through all the tissue fluids and back into the lymph, sometimes circulating around and around in this circuit for months or years. All the different lymphocytes that are capable of forming one specific antibody or T cell are called a clone of lymphocytes. That is, the lymphocyte in each clone are alike, derived originally from one or a few early lymphocytes of its specific type. Origin of the many clones of lymphocytes. Only several hundred to a few thousand genes code for the millions of different types of antibodies and T lymphocytes. How is it possible for so few genes to code for the millions of different specificities of antibodies or T cells? The whole gene for forming each type of T cell or B cell is never present in the original stem cells from which the functional immune cells are formed. Instead, there are only gene segments, actually hundreds of such segments, but not whole genes. During pre-processing of the respective T and B cells, lymphocytes, these gene segments become mixed with one another in random combinations, finally forming whole genes. Because there are several hundred types of gene segments, as well as millions of different combinations in which the segments can be arranged in single cells, one can understand the millions of different cell gene types that can occur. For each functional T or B lymphocyte that is finally formed, the gene structure codes for only a single antigen specificity. These mature cells then become the highly specific T and B cells that spread to and populate the lymphoid tissue. Mechanism for activating lymphocyte clones. Each clone of lymphocytes is responsive to only a single type of antigen or to several similar antigens that have almost exactly the same stereochemical characteristics. The reason for this is the following. In the case of the B lymphocytes, each of these has on its cell surface membrane about 100,000 antibody molecules that will react highly specifically with only one type of antigen. Therefore, when the appropriate antigen comes along, it immediately attaches to the antibody in the cell membrane. This leads to the activation process described in more detail subsequently. In the case of the T lymphocytes, molecules similar to antibodies called surface receptor proteins or T cell receptors are on the surface of the T cell membrane, and these are also highly specific for one specified activating antigen. An antigen, therefore, stimulates only those cells that have complementary receptors for the antigen and are already committed to respond to it. Role of macrophages in the activation process. Aside from the lymphocytes in lymphoid tissue, literally millions of macrophages are also present in the same tissue. These macrophages line the sinusoids of the lymph nodes, spleen, and other lymphoid tissue, and they lie in opposition to many of the lymph node lymphocytes. Most invading organisms are first phagocytized and partially digested by the macrophages, and the antigenic products are liberated into the macrophage cytosol. The macrophages then pass these antigens by cell-to-cell contact directly to the lymphocytes, thus leading to activation of the specified lymphocyte clones. The macrophages, in addition, secrete a special activating substance, interleukin-1, that promotes still further growth and reproduction of the specific lymphocytes. Role of T cells in activation of B lymphocytes. Most antigens activate both T and B lymphocytes at the same time. Most of the T cells that are formed, called the T helper cells, secrete specific substances, collectively called the lymphokines, that activate the specific B lymphocytes. Indeed, without the aid of these T helper cells, the quantity of antibodies formed by the B lymphocytes is usually small. This cooperative relationship between helper T cells and B cells will be discussed after describing the mechanisms of the T cell system of immunity. So, dear viewers, this is the end of part 3 of the 7-part series on lymphocytes and immunity in the Medicine for Anyone in the YouTube channel. Thank you for watching the video.